Okay, well, we started with our next webinar for the winter. This one's on dairy and beef internal parasites and integrated parasite management. Dr. Bryony Sands from University of Vermont is going to speak to us again this week. If you weren't in the last one, she is the IPM um, livestock extension specialist for Vermont and does a lot of pasture ecology work. I'll let her explain a little bit more about what she does and again, thank you for all for coming. So
Thanks, Ken. Yeah, hi everyone. My name's Bryony, and yeah, I work on integrated pest and parasite management in the pasture ecosystem. So, looking at sort of grazed pasture environments, internal and external pests and parasites that we find there, alongside beneficial invertebrates that we find there too, such as dung beetles. So today I'm going to focus on internal parasites. If you're interested in external parasites, lice, ticks, flies, etc., you can watch last week's webinar on YouTube, I think. Um, so this is just a quick overview. I will go over the main internal parasites that we find for cattle in the pasture ecosystem. We'll have a quick recap on IPM and beneficial insects, but with more of a focus on IPM for internal parasites this week. And then for the last section of the talk, I will show you some of our research results investigating grazing management, milk production, and young stock management in relation to internal parasites on pastures. So these are the main pests and parasites for cattle that we find out on pastures. As I said last week, we discussed the external stuff, so the stuff on the right. This week we're going to be focusing on these, so gastrointestinal worms, liver fluke, lungworm. Not going to talk about meningeal worm because that's more for small ruminants, but we will discuss coccidia. And as you can see, these pests and parasites have part of their life cycle that's completed out on the pasture, generally through the dung. But there can be other hosts such as snails and deer, as we'll find out later in the talk, that transmit these on our pastures as well. So firstly, looking at gastrointestinal nematodes, that's just worms, basically, parasitic worms. So the adults will reside inside the cow. They will lay their eggs, which are passed out in the feces onto the pasture. The eggs will hatch. The larvae of the parasitic worms will develop in the manure, and then they will crawl out onto the surrounding grass as the infective stage and sort of crawl up the grass blades and get eaten by the cow, which is then reinfected. So that's how the life cycle works. It's very dependent on moisture. So these worms and the larval stages need moisture to live, to develop, and also to move. So just bearing in mind throughout this whole talk that sort of environmental conditions and weather is a major factor influencing these transmission cycles. Generally, the wetter and warmer it is, the faster and more successfully worms, etc., can complete these transmission cycles. So the main worms that we find for cattle are Ostatagia. This is a worm that lives in the abomasum, so the fourth stomach of the cow. It is quite pathogenic, so it can damage the gastric lining and lead to some quite nasty symptoms. Cows can get immune to this. They do get natural immunity building up, but it takes a little bit longer. We normally say it takes 2 grazing seasons to build up immunity to Ostatagia. The other main one is Couperia oncophora. The adult worms of that live in the small intestine. This one is less pathogenic, so it's slightly less impact on the animal, but it can be seen in quite high numbers. And again, cattle can get immunity developing to this, and it takes a shorter time, so just one grazing season really. So looking at when these worms might pop up over the year, both of these worms have a direct life cycle, so they don't need any other animals. They just get transmitted when there's cattle there. And obviously they are around in the grazing season. That's what we generally expect when we think about parasitic worm control, is that it's a problem of the grazing season. So after cows have been turned out in the spring, there's normally a 3 to 4 week delay, and then they can start picking up these worms and you can start seeing issues. Another thing to note, however, is that both of those types of worms can overwinter inside the animal. So they will go into stasis as larvae inside the gastric lining, for example, of the cow. And then they can sort of have a massive emergence from the gut lining, and this can be very damaging and it can be fatal. And we see that in the winter, and that's called type 2 disease. So grazing season is type 1 disease, and then this case where we have overwintered larvae inside the animal is type 2 disease. So I just wanted to mention that because we do need to be looking out for things like worms, even in the winter. So young animals are mostly susceptible to these worms. As I mentioned, older animals get natural immunity. Generally, signs of infection are reduced appetite, reduced feed intake, and scours, sort of, rough hair coat and just a loss of condition. So you can see this young animal here has got a mucky behind, so that's a good indication that there's something going on. Another thing to note is that subclinical infections, so that's low levels of worms that aren't causing these sort of signs of infection, can still have impacts on weight gain, on milk production, and on calving rate. So it is important that we sort of get this under control. So moving on to liver fluke. Liver fluke has an indirect life cycle, which basically just means it needs another host as well as the cow in order to complete its transmission cycle. And the intermediate host, so the other host for this parasite, is the mud snail. So snails obviously need moisture, and that means that this particular parasite, there's a much higher risk in sort of wet pastures, damp areas, places where there's standing water. That is a major risk factor for liver fluke. The adults live in the liver bile ducts, and all ages of cattle can be susceptible to this. So immunity is not really acquired in the same way as those other worms I mentioned. And so the juvenile stages of the liver fluke can be acquired, well, they are acquired over the summer and in the late fall, and we can see clinical signs from that. The adult worms take quite a long time to develop inside the cow. I think it's about 8 to 12 weeks. So we do also see disease from liver fluke in the winter when the adult worms have developed inside the animal. So that's just highlighted on this calendar here again. Acute fluke is when we see problems from those juvenile stages in the late summer and the fall. And then chronic fluke is when we see problems from the adult worms, and that is generally into the winter. So there can be minimal symptoms from liver fluke until it's quite developed. Symptoms include weight loss and poor performance, bottle jaw and sort of swollen brisket is a sign, as well as scours. But often abattoir reports and condemned livers at slaughter can be the first thing that we know about having liver fluke in the herd. But it's important to keep on top of this and to be aware of it because it can actually kill animals. I had it in my goats last winter, and I really didn't see any symptoms until it was too late, and animals sort of died within a day of showing symptoms. So this is something that we really need to be aware of if we have wet pastures, thinking about what we can do to prevent this. And the liver fluke are quite nasty, so they also transmit these other diseases. They will sort of, as they're kind of swimming around the liver, they track clostridial bacteria around, and that can cause something called black disease, which leads to sort of necrosis of the liver, and also red water disease. Just wanted to show you some pictures here. This is from one of my goats that passed away, and when I dissected the liver, I could see all this damage and these there's actual fluke in here. So this is a liver fluke that I pulled out. And then this is a picture of a severely damaged cow liver. So as I said, it can cause significant impacts, but we don't necessarily see the signs of that straight away. Okay, so moving on to lungworm. Lungworm has a direct life cycle again, so it's just the cow that transmits this. And the adults will reside in the lungs, and they will lay their eggs in the lungs. The eggs will develop into larvae, which will get coughed up, and then the cow will swallow them. And the larvae then pass out in the dung and complete the rest of their life cycle to the infective stage on the pasture, where the cow eats them and reinfects themselves. So this is also very dependent on moisture and damp conditions, and disease usually presents later in the summer or the fall. So the clinical signs of lungworm is really the coughing. That's a giveaway for this. Respiratory disease can also be caused by a general loss of condition and reduced production again. And young stock are generally the most affected. Animals can acquire immunity. It's important to note that some adults can also be infected, and there's this sort of hypersensitivity response where cows that haven't quite built up immunity to this, when they encounter it for a second time, they can have this quite strong response. So we do need to look out for it in older adults occasionally as well. Diagnosis is slightly more difficult because, as you saw, it's not the eggs that are passed out in the feces. So our common tests like fecal egg counting wouldn't show this up. But then again, as I said, cows will generally be coughing. And if we're seeing that, there are these other tests where we can look for larvae in the feces or doing blood tests, for example. So the last one is coccidia that I'm going to mention today. Coccidia is a protozoan parasite, so it's not a worm. It's basically a single cell, and it lives inside the cells of the intestinal lining of the cow. And because it sort of lives inside those cells, when it multiplies in the cow, it can really damage the lining of the intestines. Again, this is fecal oral transmission, so it's transmitted through the dung on pastures and in housing as well, as we'll see. And it can have quite a quick transmission cycle of just sort of 2 to 4 weeks, so it can really proliferate quite quickly. This is mainly an issue in calves, so we see calves affected in the spring and early summer after they've first been turned out to pastures. But we also see this in the winter in sort of overcrowded housing, or where there's calves that are sort of in groups and there's lots of dung around. So this can happen at any time of the year, really. And one of the main risk factors out on pastures is contaminated water bodies. So, standing pools, little ponds, permanently wet areas, they should really be fenced off because animals sort of pooping into that can really be a breeding ground for this. And then crowded areas, obviously, sort of around water troughs, etc. Symptoms in calves are very strong smelling and profuse scours, generally with mucus and blood in, and then wasting and a poor appetite and a dull coat. So that is the main internal parasites. And this is an IPM webinar, so I'm not going to let you get away without me mentioning beneficial pasture insects. So this is just a quick slide showing the dung ecosystem. So obviously those parasites that I've just gone through generally depend on the dung to complete their life cycle out on the pasture. So in the manure, we have sort of pests and parasites transmitted, yes. But I also just want you to be aware that there's also this whole host of other beneficial organisms that we find in manure. So it's not all bad. We have coprophagous insects, which will eat manure and sort of live there and breed in it. And they are very important in dung decomposition. So recycling all of those nutrients and organic matter back into the soil, helping with pasture fertility. And then we have other organisms that are predatory, for example, mites, which are known to predate things like parasitic worms. So they are really important in providing a natural suppression of pests and parasites on pastures. So I do just want you to bear in mind when we're going through this webinar that there are things in the manure that we want to be protecting, as well as the pests and parasites there. So I will just do a quick recap of IPM and beneficial insects. Some of these slides, like a couple of them are a bit similar to last week. I want to make sure that people who didn't see that information get that now so that we're on the same page. And there's a lot of new information in there too. So if you were watching last week, don't zone out. And then I will talk about IPM with more of a focus on internal parasites. So clinical disease from these parasites is usually avoided by using various veterinary treatments. Those chemicals are very good at controlling parasites as long as there's not resistance in the parasite population. But there are major issues that we are now finding out with the common treatment strategies and sort of the overuse of these chemicals. So firstly, that is the development of resistance in parasites, leading to drug failures. So if you think about regularly treating your whole herd, that's going to apply a strong selection pressure on those parasites to evolve resistance to the treatment. It's sort of the same idea as with antibiotic resistant, and why your doctor will tell you not to take too many antibiotics. We don't want resistant bacteria popping up. We are seeing resistant populations of worms, of livestock, and treatment failures sort of globally now. So it's important that we conserve these chemicals for when we really need them. Secondly, reduced immunity in our livestock. So as you saw on those previous slides, cattle will develop natural immunity to these parasites. But in order to do that, they do need some level of exposure. So they need to be exposed to low levels of parasites when they're young in order to build up that protection for life. So if you're sort of not allowing any contact with parasites, you're really setting yourself up for more problems with animals when they're older because they haven't got this natural immunity. And there's things that we can do so that we can carefully ensure that animals are building up immunity. And I'll talk a bit about that later on. Finally, negative environmental impacts. So, as I mentioned, these chemicals are very effective at killing pests and parasites. They're usually insecticidal or wormicidal. However, when we treat cattle, they come out the other side and they're still insecticidal when they come out in the manure. So we know that up to 90% of the dose can be excreted largely unchanged, so still insecticidal. And obviously, if we're having insecticide residues in those dung patties on our pastures, that's going to have negative impacts on those beneficial insects that I was speaking about. And I wanted to do a study, this was about 10 years ago now, but just looking at what the potential of those beneficial insects, such as dung beetles, was in controlling parasites on pastures. So we collected almost 900 pounds of cattle dung that was naturally infected with some of these parasites. So these are the ones that we went through earlier, Cuperia and Ostatagia, the main gastrointestinal worms. We mix it all up with a cement mixer. And then we did a field study where we laid out all of these artificial dung patties on a pasture using this manure that we know has parasite eggs in. So we basically wanted to see if dung beetle colonization on those patties could help to reduce worm transmission when those eggs would hatch and the larvae would crawl out onto the grass. Can the action of the dung beetles help to reduce that? So we had 3 treatment groups. One of them was the dung patties had no dung beetles, and we achieved that by putting these cages over. So there's a dung patty in there, and that's protected from any dung beetles getting to it. Then we had patties that were naturally colonized with dung beetles and ones that had enhanced colonization. So we added extra beetles to those ones. And then we cut the grass around those patties every 2 weeks for 10 weeks over the grazing season to extract those infective worms from the grass around it. So in this bird's eye view, you can see where we've been cutting the grass around those patties. So looking at the results of this, it looks quite complex, but I'm going to walk you through it. So hopefully it'll be clear by the end. This figure is showing the number of gastrointestinal worms, so the infective worm larvae that was on the grass around those dung patties. And then along the bottom, it's the week, so how long the patties had been out on the pasture. And then the different bars represent the patties with different levels of dung beetle colonization. So the white ones have no dung beetles in, the light grey had natural levels of dung beetle colonization, and the dark grey had extra dung beetles added. So when we look at the first 2 weeks, you can see the dark grey bars there actually had more parasite worms on the grass around it. So the more beetles equals more parasite larvae at week 2. And that can be explained because those parasite eggs basically need oxygen in order to hatch. And we think that the dung beetles colonizing those patties and sort of aerating it and tunneling it provided more oxygen and allowed the parasite eggs to hatch. So there was more larvae there. Looking at from weeks 4 to 6 of the experiment, there was no difference in the number of parasite larvae around the dung patties with different levels of dung beetle colonization. However, when we get to week 8 onwards for the rest of the experiment, you can see here that the dark grey and the grey bars, so the ones with more dung beetles, had significantly lower numbers of parasite worm larvae on the grass around them. So basically, over the course of this whole grazing season, dung beetles were reducing the number of parasite larvae coming out of those dung pats. And that is likely because the action of those beetles over that grazing season, sort of fragmenting and tunneling and breaking down the dung, would completely dry it out or remove it, taking away the breeding ground for those larvae, whereas the ones with no dung beetles were kind of intact, and those dung patties could keep pumping out parasite larvae onto the pasture. So the conclusions from that is that over the summer grazing season, we did see a 20 to 30% reduction in the parasitic worms on pastures as a result of the beneficial dung insects, so the dung beetles. Important to note that high rainfall events overrode that. So when it rained heavily, there was a load of parasites, no matter what. So again, just thinking about sort of how the weather can really affect the risk of this. Okay, so back to the IPM techniques. This slide is just showing you what common practice might look like that is not based on IPM management. So this is based on calendar treatments throughout the year. So if you just have a look at, I've put all of these different internal parasites that we went through and when they could be popping up over the year. And these treatments are what might be given preventatively, just based on thinking that we may expect to see problems and we want to prevent that before it happens. And generally, this would be treating the whole herd with these treatments. So this can be perceived as sort of the lowest risk and the lowest cost method. However, we will see that that is not necessarily true, especially when we consider parasite biology. So we know that about 80% of parasites are generally found in just 20% of the herd. So that means most of the parasites are actually in a small number of animals. And by treating the whole herd regularly like this, it could really be seen as a bit of a waste of time and money because not everyone necessarily needs treating. So solutions in terms of an IPM approach is really based on finding out if there actually is a problem and which animals need treatment. So the motto for this is test, don't guess. And we can do various monitoring diagnostic tests, which I'll talk about in a minute, to find out what level of issue we have, if there is one, and who we need to be sort of treating, if anyone at all. There's other things we can do preventatively here, like vaccination. So there is vaccinations for lungworm and for clostridial diseases that are transmitted by fluke. So that can really help to prevent problems from popping up in the first place. So this is based on treating only when it's actually necessary instead of just preventative treatment like in the previous slide. We can use less harmful products. So if we just take a look at this, this is a sliding scale of different products. And we know that some products are more environmentally damaging, so they have more toxic impacts on dung beetles, for example, than others. You can see here that ivermectin and eprinomectin, unfortunately, are known to have quite toxic impacts on dung insects. But there are other choices we can make as we go down the scale here. You can see moxidectin, so that's cydectin, and that is known to have fewer toxic impacts on the environment. So we can make sort of different treatment decisions based on knowledge about that. Rotating products is obviously a good thing to do to help to slow resistance. And then really a cornerstone of IPM for internal parasites is good grazing management. And we'll discuss that in just a moment. So looking at these IPM tools for monitoring and diagnostics, as I mentioned, that's looking for the presence of infection. So is there actually a problem? And the severity. So yes, we're seeing maybe eggs in the feces, but is that at a level where we need to do something? Or is it at a level that's acceptable to us? So fecal egg counting is a really commonly used fairly straightforward and fairly cheap way of finding out if you have an internal parasite problem. I'll go into a bit more detail on that in a slide in a moment. Then we can do body condition scoring as well to see if animals are losing condition. And then there's these other tests, so blood or milk tests that can pick up whether we have internal parasite problems as well. Fermature, you might have heard of, that is used in small ruminants, so I won't go into too much detail, but it's basically looking at the whites of the eye to see if there's anemia that's caused by worms. And then considering treatment thresholds, so these tools can show us sort of the level of infection, and that is important because grazing animals will have parasites. That's just what happens when animals are sort of out there eating grass. So it is likely that animals will have internal parasites, but we need to know at what level that actually becomes an issue and at what level we need to take action. And hopefully we are avoiding a level where we're seeing clinical signs such as scours. Obviously that does happen, but by using these tools, we're trying not to get to that point. So internal parasite IPM practices, as I mentioned, good grazing management is foundational to this. A key idea here is maintaining a high enough sward height. So as I explained to you, most of these internal parasites need a moist environment. They need a film of moisture to sort of move around in. Otherwise, they dry out and die. So it's known that sort of worm larvae, for example, will only be found in the bottom sort of 4 inches near to the soil where it's moist. They won't climb up much higher than that because they risk dying. So if we are making sure that our animals are not grazing down to that bottom 4 inches, we're hopefully avoiding them even coming into contact with these parasites. So a key way of doing this is obviously using rotational grazing, moving animals frequently onto a new pasture, onto a clean pasture, allowing pastures to rest and have enough time to recover. Obviously, that's good for sort of forage health and production, but also breaking these transmission cycles of internal parasites on our pastures. Allowing animals to gain their natural immunity, as I mentioned. Mixed or sequential grazing. So you can see here that there's sheep grazing with cows. And that works because most of these internal parasites are very host specific. So cattle worms will not infect sheep generally, and vice versa. So if a sheep comes along and bites a piece of grass that has cattle worms on it, those worms will die inside the sheep. They can't be transmitted. So by doing sort of mixed or sequential grazing, we're helping to break transmission cycles and keep numbers down. There's high tannin foragers that you might have heard about. So those are things like bird's foot trefoil, chicory, sainfoin, small burnet. They contain chemicals called tannins, which are naturally sort of anti-parasitic. So they help livestock to be resilient to these parasites naturally. And then obviously our biological control by supporting beneficial insects on pastures. So I just wanted to show you, this is a good tool for making sure that we are not exposing our vulnerable animals to very high risk pastures, but we are allowing them to come into contact with some level of worms. So this is called pasture risk planning. This is an example here that's been based on sheep, but it applies to cattle as well. So if we take a look at a low risk pasture here, that might be one where there was no cattle grazing last year. It might have been arable last year or hayed. So it's had a whole year off, basically. We know that there's likely to be very low numbers or no parasites on that pasture. Looking at this medium risk pasture, so if we graze older animals, especially dry cows, for example, they're pretty resilient to internal parasites and they're not likely to be shedding and transmitting many at all. So a medium risk pasture might have had dry cows earlier in the year, or the previous season. It might have also been hayed earlier the same year. And then if we look at high risk pastures, that's mainly pastures where we've had young stock. So younger animals are, as I mentioned, much more susceptible. They're much more likely to be spreading parasites around. So things that we don't want to do is put our vulnerable animals and our young animals on the same pastures each year. So by knowing the risk of each pasture, we can make good decisions about which groups of animals to put there. And this is a nice example of a farmer who's sort of just done that up for himself. Okay, so going into a bit more detail about fecal egg counting. This is, as I mentioned, quite a straightforward and cheap method that can be used to detect problems early in the herd so that we can keep on top of problems before they get out of hand. It can also be used to diagnose animals that we suspect do have an issue. And then it can also be used to detect resistance to treatments in worm populations on our farms. So the fecal egg count reduction test is basically doing a fecal egg count and then treating with our product and then doing another fecal egg count a couple of weeks later to see if it actually worked. So if we're not seeing sort of at least a 90% reduction in the number of worm eggs after treatment, we know that we have some level of resistance to that particular treatment and we need to switch to a more effective treatment. You can get this done through your veterinarian. There's lots of commercial lab services that do this. You can also do it yourself at home. I know a few farmers who have got microscopes now, and this kit is fairly inexpensive. And once you know how to do it, it can be quite a valuable tool to just do this yourself. It gives you real-time information so that you can make treatment decisions much quicker than if you had to sort of send the sample away and wait for the results to come back. So this just shows what a slide might look like. And you can see these larger eggs here are the gastrointestinal worms. And then we have little small oocysts here, which is coccidia. But if you're interested in learning more about this, I do have some resources that I can send you. So feel free to email me and I'll send you some more information. It's important to note that fecal egg counting is not always useful for everything, and that's because not all of these parasites will be shedding eggs in the feces. So we shouldn't be relying 100% on this. For example, it doesn't work for those type 2 worm infections. So remember I said we can get these gastrointestinal worm issues in the winter, and that is from the larval stages, so they are not producing eggs. Liver fluke takes sort of 8 to 12 weeks to develop into an adult inside the animal. So a lot of that time it's a larvae and not producing eggs. And then lungworm, as I mentioned, that is the larvae that comes out in the manure. So obviously fecal egg counting won't pick that up either. And so if you suspect any of those issues, it's best just to ask your vet for what testing you can use to find out. There are lots of options, so these blood or milk tests and then special tests for lungworm as well. So I did just want to mention treatment thresholds for fecal egg counting. I want to just have a big caveat here that each farm and each animal is obviously different. So it's important to take these with a pinch of salt and look at them in relation to your specific goals on your farm and specific animals as well. So for cattle here, generally anything below 100 eggs per gram is considered to be not really an issue. We have between 2 and 700 eggs per gram. We might want to think about doing something depending on the situation and depending on whether the animal has clinical signs or not. We always want to be considering whether the animal is showing signs, because some animals are very resilient and they can have a high egg count without it really bothering them, whereas other animals will be very affected by lower egg counts. When we get to sort of 1,000 eggs per gram, it's likely that we need to do something about that. Coccidia, again, we really have to be looking at signs and symptoms as well as these oocyst counts. And that's because there's a lot of coccidia species which are not actually pathogenic. So we might have a high oocyst count in the fecal egg counting slide, but it might not actually be a pathogenic species. So we always say that when diagnosing coccidia, it has to be these egg counts alongside signs and symptoms too. So all of these techniques are helping us to achieve something called targeted selective treatment. And that is basically treating specific individual animals when there's evidence that they actually need it, instead of those blanket treatments of the whole herd that we saw earlier. And that can help us to use as little product as possible, but as much as necessary to make sure that our animals are healthy. So benefits of this is that it slows resistance in worm populations. And that's because it provides something called refugia animals, which basically just means untreated animals. So the worms inside those animals will not be developing resistance because they haven't been exposed to the treatment. Whereas other worms in your herd from treated animals will be developing resistance. But because those worms will be mixing and breeding, it sort of dilutes the development of resistant genes and helps to slow development of resistance in that worm population on your farm. Secondly, this will obviously protect beneficial insects such as dung beetles by providing refugia dung pats. So that's, again, just untreated, non-toxic dung patties. They'll be around at all times, so not all beneficial insects will be exposed to insecticide residues. Okay, so for the last section of this talk, I'm going to just go over some of the results of our research, looking at grazing management and milk production and young stock management in relation to internal parasites. So this is a study that we did in 2022. We worked with 29 grazing dairy farms in Vermont and New York State. And these farms were practicing different grazing strategies. So they were either using continuous grazing, rotational grazing, or management intensive rotational grazing. And within those different grazing strategies, we had farms that were using chemical worming treatments and farms that were not using any treatments. And on those farms, we measured internal parasites. So we did fecal egg counting on the manure samples from the herd. We looked at pest flies. I won't talk about that today, though. We also looked at soil health outcomes using Cornell soil health analysis. And then we monitored beneficial insects such as dung beetles as well. So this is the results of the internal parasites. This up the side here is the fecal egg count, so the number of worm eggs that were in the manure samples. And then along the bottom, we have the grazing strategies that the farms were using. So continuous grazing, rotational grazing, and management intensive rotational grazing. And then the blue bars are farms that were using no worming treatments at all. And the pink bars are farms that were using worming treatments. So we can see that for farms that were continuously grazing, if they were not using any worming treatments, they had significantly higher risk of internal parasites in their herd, significantly higher fecal egg counts there than the farms that were using worming treatments. However, when we look at these rotational grazing strategies, we can see that the farms that were using no treatments had significantly reduced risk of internal parasites in their cattle down to a level that was equivalent to the farms that were using worming treatments. So that just shows that rotational grazing strategies can effectively control internal parasites of cattle down to a level comparable to farms that were using chemical pesticides. I think I'm just going to skip over these two slides. These are basically showing that dung beetle abundance on the pastures help to reduce pest flies and also internal parasites. And then dung beetles also help to improve soil health outcomes and reduce soil compaction. But I don't really have time to go through that in detail. The key messages from this study were then that rotational grazing strategies can effectively control cattle internal parasites. Dung beetles were naturally reducing pests and parasites on pastures and were contributing to improved soil health outcomes. So in conclusion from that, we can see that good grazing management can contribute to effective pest and parasite control, whilst also supporting beneficial insects and soil health. Okay, so moving on to the final study that I'm going to show you. This was looking at grazing management and milk production, and we did this just in the grazing season gone, so 2025. And we worked with 8 grass-fed dairy farms in Vermont and New York State. And we went to those farms every month to take fecal egg counts from the lactating herd, and we correlated those egg counts with grazing management practices. We also went to farms on milk test day so that we could take individual fecal egg counts and correlate individual animals' parasite burdens with their milk production. So this is showing parasite burdens in relation to grazing management, and this is post-grazing sward height. So that's the residual that I was talking about. So how much grass was left after the animals had finished grazing that pasture. So this is showing the number of worm eggs in the feces up the side and that post-grazing sward height along the bottom. And it's showing a significant negative relationship between the number of worm eggs in the manure and the post-grazing sward height. So basically, cows grazing pastures down to a lower sward height had more worm eggs in their feces. And that is just highlighting the importance of keeping that at least 4-inch residual so that animals are not coming into contact with all of those parasites. Looking at parasite burdens and milk production, again, up the side, we have the number of worm eggs in the feces. Along the bottom, we have the milk yield of that animal. And again, there's a significant negative relationship here between the number of worm eggs and the milk yield. So cows that had more worms produced less milk. And that, again, really highlights that good parasite control really matters. Even at subclinical levels. So these are quite low levels of worms. We wouldn't really be seeing any symptoms such as scours, but even at these low levels, it is impacting milk production. Other notes were that cows in the earlier stages of lactation had significantly more worm eggs and coccidia in their feces. Obviously, the nutritional demands of sort of calving and beginning lactation means that less resources are given to the immune system. So that just shows how important it is to be supporting those animals nutritionally. And then also milk from cows that had more worms also tended to have a lower fat content, and those animals had higher somatic cell counts. All right, so looking at young stock management in relation to internal parasites, these were the same 8 grass-fed dairy farms in Vermont and New York State. We did monthly visits to those farms as well and took fecal egg counts from the young stock. And we correlated those egg counts with grazing management practices, milk feeding quantity and duration. So how long did they that farm feed milk to their calves and how much milk was fed, and then also different calf rearing systems. So this shows parasite burdens in relation to grazing management in the young stock. We have the number of worm eggs up the side again, and then the pasture occupancy period along the bottom. So how long were those animals grazing in that particular pasture for? And this shows that young stock grazing the same pasture for more days had significantly more worm eggs in their feces. Except for these very long periods, which was kind of a surprise. They were more like continuous systems. And as I mentioned, it was this grazing season just gone. It was incredibly dry, and those farms were just feeding hay at that point. So I think that's why we were seeing that. But generally, we're seeing the longer that you leave those animals in the same pasture, the more risk there is for acquiring worms. And we have this sort of cutoff here where we say maximum of 3 days grazing on one pasture. This is showing the same thing, the pasture occupancy period, but this is for coccidia this time. And this is showing that young stock grazing the same pasture for just 1 day had significantly lower coccidia than those higher occupancy periods. So occupancy period really matters for coccidia as well. Looking at, this is milk feeding duration of those calves. So here we have the number of worm eggs in the feces, and then how many months were those calves fed milk for? And this is a significantly negative relationship again. So calves that were fed milk for longer had fewer worms. So that is just highlighting that trying to extend milk feeding time can be very beneficial for calves building up resilience to parasites. Okay, and then this is the final thing showing oocysts of coccidia up the side. And then we have, were those calves raised in a group feeding system or not? And we can see that calves raised using group feeders had significantly more coccidia than calves that were not raised with group feeders. So that's just highlighting the importance of hygiene relating to sort of calves being in groups and the risk associated with the manure spreading coccidia around. All right, so key messages from this were that grazing pastures down shorter led to increased gastrointestinal parasites, so increased worms. Cows with more worms had reduced milk production, even at low subclinical levels. Moving young stock to a clean pasture frequently, so hopefully 3 days or less, is important for worm and coccidia management. Feeding calves milk for longer protected them from internal parasites, and group feeding systems for calves may be increasing coccidia risk. Right, I think I'm just about in time. It was a bit long, but I'm happy to take any questions if there's time.
Thank you, Bernie. That was really, really excellent. We do have several questions in the chat here. So the first one is, what triggers the emergence of larvae from the gut lining in late spring or late winter and spring?
That is a great question, and one that I cannot confidently answer. I think it's related to the sort of the weather conditions. They generally go into this hyperbiosis state in adverse conditions. So when things start improving in the spring, they will sort of emerge in order to complete their life cycle.
Okay, and just one note, there is a link there to do a survey on the webinar. If you could take that, that would be great. Just gives us feedback on what we did well and what we can improve on, at least relative to the transmission of the webinar. So I did answer this question, what is an IGR? But then they went on to ask another question. How long does the insecticidal effects of different toxic dewormers last in the manure? Say if I were to spread manure on a field, how long would I have to wait to be sure it wasn't negatively affecting the impact that field on that field?
Yeah, so that really depends on the weather conditions. A lot of these compounds, like ivermectin, for example, degrade quite quickly in sunlight. So if it's summer and it's hot, it could be sort of 3 to 4 weeks. We have done studies looking at manure with these residues in over winter, and we found that the residues were still there 4 months later. So I would say, in sort of cooler conditions where there's not much sunlight, it can be many months, whereas in midsummer, it's a bit safer.
Okay, then the next one, would you recommend buying dung beetles to introduce to your pasture? If so, do these beetles need to come from the Northeast?
Yeah, that is a question we get a lot, and I would say that the most important thing is really getting the conditions right in order to support dung beetles that are already in your pastures. Um, Particularly, I think that where people can buy dung beetles from is not necessarily companies that are in the Northeast. So also some of those species may not even survive after you release them. And again, if you're using some of these products regularly and then releasing beetles, it will probably negatively affect that. So yeah, so I think my advice would be fostering your own natural dung beetle populations first.
The next question is, where can you purchase these egg counting kits?
You can get them online easily. The one that I had on that slide was FEC source. You can get that on Amazon and it's really cheap. You can also get a microscope fairly cheaply. From that same company does microscopes as well, actually. But there are lots of different companies, and a microscope is probably I think it's around $200 to $300. There's probably cheaper ones, but overall, the cost and the availability of this equipment's pretty good.
How often would you recommend taking fecal egg counts for monitoring?
I would start a couple of weeks after, maybe 3 weeks after you've turned animals out, and then probably every month for a general monitoring over the grazing season. If you Obviously, if you're suspecting issues, you might want to do individual animals or things more frequently. But yeah, I would say once a month over the grazing season.
Are there any other questions? Oh, here we go. How often do you recommend taking, oh, sorry. The graph from the dung beetle research showed that larvae populations were still high at 8 to 10 weeks in without dung beetle activity. How does that compare with grazing rotations that are shorter?
Yeah, so this experiment was carried out in the UK. So our summers are much cooler than in the Northeast. So, for a grazing rotation in a cool and wet climate, you'd really want to be leaving those pastures for 60 to 80 days to try and break the transmission cycle because those larvae last for so long. Somewhere like the Northeast, in a hot and dry summer, you can probably get away with sort of 30 to 40 days rest to kill off the worms and break those transmission cycles. But it's very weather dependent, and the infective larvae are incredibly resilient and can last for months and months when it's cool and moist.
Great. And Hannah put the source of the egg count kits. Hannah, could you also put that link back in for the evaluation? So if you could, before we end this, if you could click on that and take that little survey for us, it'd be great. If there's no other questions, we're right at 1 hour and 1 minute, so or two. So we hit it just about right. We are going to post this on YouTube, our Cornell IPM YouTube page, and we posted last week's as well if you happen to have missed it. So again, Bryony, that was a great talk, and thank you very much.
Thank you.
All right, thanks everybody.